Thanks, Anna. Hello, everyone. Thank you all for joining. I'd like to first apologize. I have a bit of a cough, so please bear with me. I have a cup of tea here with me, so I hope I'll be able to keep it under control. So for this webinar, I'll be giving you all a broad overview of different metagenomic and bioinformatic approaches we can use to better understand the role of the human microbiome in health and disease. So, as a bit of an introduction, one of the fascinating things of working with microbes is that they're essentially everywhere. So, wherever we look, wherever we are, there are microbes everywhere. We have them in the ocean, in the soil, in animals inhabiting all sorts of environments, things like Antarctica or even, traditional tropical environments. We have microbes in hot springs, in mountains. So, we have microbes that have adapted to various different environments and developed their own unique adaptive traits. Now, in the case of the human microbiome, this has become quite a hot topic in recent years. And one of the main issues and challenges of working the microbiome is how diverse it actually is. So the human gut microbiome in particular, which has the highest density of microbes in our bodies, is colonized by over 40 trillion microbes that span all domains of life. So we have bacteria, archaea, eukaryotes, and viruses in there. And what has been shown so far is that the gut microbiome has many important health benefits. So it can help us with digestion, specifically in the digestion of certain fibers that we ourselves are not able to metabolize. It can help us with immune regulation and also against protection against external pathogens. So the microbiome itself can act as a barrier that prevents important opportunistic pathogens to kind of invade and cause disease. But the microbiome is highly diverse and unique. So every one of us has their own unique microbiome, especially when you consider things like strains and species. We all have a very unique composition. So trying really to understand how that composition relates with different aspects of human health and disease has been one of the main focuses and challenges in the field. So far, lots of studies have been finding associations between the microbiome and different health states, things like colorectal cancer, obesity, inflammatory bowel disease, etc. And even there are new studies even suggesting that the microbiome may go beyond typical gut-associated disorders, things like neurological disorders, physical fitness, and even our behavior. So there have been huge develops in the human microbiome field. And if you look back to almost 80 years ago, you can definitely track a huge development, both in terms of our knowledge of the microbiome, but also in the methods and the techniques that have been used. So in the early 20th century, really one of the main things that kind of developed the field was the ability to start experimentally testing many of the microbes that exist in our intestinal tract. Now we know that these microbes are mostly strict anaerobes, so they do not survive in oxygen exposure. So having developed that ability to then culture these microbes has been one of the main points in the field. There have been developments in studying fecal transplantation, so what impact transferring an individual's microbiome to another individual would have on things like Clostridium difficile infection, developing germ-free mouse models, studying drug metabolism in the gut. And even in the 80s, this is really when we started to look at the microbiota in early life, thinking of how about the species that colonize infants, what is their impact on the development of these individuals. But then, for the purpose of this talk, one of the main kind of revolutionizing aspects was the development of DNA sequencing of the microbiome and ability to study the microbiome stability and individuality using these methods. So this graph, I'm sure many of you working in the genomics field essentially use this graph all the time for your talks. I've seen it multiple times. So this is just showing how DNA sequencing costs have drastically reduced around the 2000s. And this was really with the implementation of things like high throughput sequencing technologies. So in the early 2000s, this essential reduction in sequencing costs led at the same time to a massive increase in the number of microbiome studies. So a lot of people were essentially using DNA sequencing to profile the microbiome and build its interaction and relationship with human health. Now, one aspect when we're talking about DNA sequencing of the microbiome, one concept that you may hear all the time is this idea of metagenomics. Now, metagenomics is essentially the analysis of all the genetic material of a sample without any prior enrichment. So, as the name suggests, it derives from genomics. So genomics is a study of the genetic makeup, the genetic material of an individual organism, whereas metagenomics, you're essentially looking at the whole community together. It's important to distinguish that as well from 16S RNA genotyping. This is also very common in microbiome studies, but 16S RNA genotyping just literally means just studying one particular gene, the 16S RNA gene, which is a ribosomal gene that is conserved across all bacteria. Now, this is a useful marker because it essentially it's conserved across bacteria, and you can use its variation between different bacteria to sort of profile and type these different organisms at taxonomic levels, such as genus or family. But it does not provide the same level of resolution as metagenomics. So metagenomics, we can have an understanding of the microbiome at the species and subspecies level, and it will give us information about the functional capacity. Whereas 16S, we're really looking only at a taxonomic marker. So to emphasize, the use of metagenomics is important both on a taxonomic front, so to actually try to type and identify each individual organism. So the DNA sequence we recover from the metagenomes will act as a unique fingerprint of every single organism. But at the same time, since the DNA is sort of a blueprint for all the proteins that that organism will produce and synthesize, we can use them also to predict the functional capacity of these organisms. We can look at the metabolism and then be able to make hypotheses about what that species is able to do. So how do we actually obtain this data? So the most common material we start with when looking at the human microbiome is a stool sample. And essentially the first step to actually recover a metagenome is to extract the DNA that is present in these stool samples. This is usually done through a series of standardized protocols using a mixture of physical and chemical steps that essentially allow you to recover all the genetic material that is present in the sample. Then essentially need to prepare this DNA in order to be read by the machine that's going to actually sequence the DNA, the DNA sequencer. And these kind of prepared DNAs are called DNA libraries. And then once you have the DNA library, you can actually sequence it with a traditional DNA sequencer, which will essentially, to boil it down, essentially take some sort of photocopy of your DNA. But what will output is the sequence fragments, what are called reads, that will essentially represent pieces of your original DNA, of the DNA that you had in your original template. Now, after you do the sequencing and when you're planning which DNA sequencing technology to use, what you really want out of it is a high yield. So you want to have lots of reads representing your sequence. So the more reads you have, the better coverage you will have of the whole genome that is present in your sample, and the more confidence you will have on how the sequence is identifying the different positions in your DNA. And then the other thing that you want are long reads. So as I mentioned, it's inevitable that due to how these sequencing technologies work, that you're not gonna get the complete genome ad. This is very rare. Only a few technologies are able to do that. So most time, what you get is just these sequence fragments. But the length of these sequence fragments is quite important. So the longer they are, the more resolution and better they will work to do subsequent bioinformatics analysis. So depending on the choice of sequencing technology, there's going to be a trade-off between the depth you're able to achieve, the accuracy you're going to have for the sequences, the cost, and the length of the reads. So typical high-throughput sequencing technologies use Illumina technology. There are different platforms that are available, but essentially Illumina is characterized by doing short read lengths, so below 300 base pairs most of the time. But the main advantage of Illumina is it provides very high accuracy. So you're able to be very confident in the different positions that are called and identified in your sequence reads. And in general, there's a lower cost per base. So given you reach very high levels of depth, it ends up being less costly if you essentially sequence multiple samples at the same time. Now, with newer technologies like long read sequencing, you can actually get longer read lengths that go beyond hundreds of kilobases. Now, the problem with that is that in general, you get lower accuracies than compared to Illumina, and that can also lead to higher costs per base because you're also going to need to sequence more to be more confident, and at the same time, it doesn't provide as much depth as a typical Illumina sequence. Now, after you essentially run your data through these sequencings, you get a huge amount of data out there. And then the question is how to actually make sense of all this. And this is kind of what I'll get into as a main bulk of this talk. So as you get your sequence reads out, the first thing you really want to do is pre-process your data. By pre-processing, I mean essentially look at the quality of the sequences and be sure that you are removing sequences that are either low quality or trimming the sequences in a way that removes low quality bases. So the way that you do this is you use tools like FastQC and TrimGalore that allow you to look at the sequences individually. And if a sequence, for instance, has specific errors on extremity, you can trim it and see if you can keep the main sequence there. Or if you have too many errors in the sequence, you just remove it altogether. Once you have your quality filtered reads, then particularly in human microbiome studies, the next thing you want to do is actually remove any possible human contamination. And you can do this with typical mapping approaches where essentially you use tools like BWA Bowtie, where essentially you're mapping the sequence reads to a template of the human genome. And then you fish out everything that did not map to the human genome. So those would be theoretically your microbial sequences. This you can do with things like SAM tools or BET tools to extract clean sequence reads, and these are the ones you're essentially going to be working in your study. Now, in terms of the methods that we can use to analyze this data, I'm going to divide the methods into two main categories. So there are assembly-based methods and then reference-based methods. So assembly-based methods essentially rely on your ability to align and reconstruct longer sequences from these sequence reads. And they're very valuable because by reconstructing longer sequences, you're able to obtain a more accurate classification of your bacteria. You're able to find new species and new genes that are not represented in reference databases. But at the same time, a few cons working assembly-based methods is that it's usually more complex and time-consuming. There's not one standard pipeline that you can use. You need to kind of consider different pros and cons of different approaches and really think about what is the question you're asking to make the decision. And at the same time, since in assembly-based methods essentially collapse the diversity of sequence reads into one consistent sequence, is usually not quantifiable. So the assembly by itself will not really give you an estimate of how much of that bacteria, how much of that gene is present there. In contrast, reference-based methods are generally faster and easier to perform, and they allow quantification of the species and functions. So since you will know how many sequences map to different species or different functions, you can use that as a proxy of the abundance of those features. The limitations of reference-based methods is that they require comprehensive databases, so you're not able to find new things that are found in your samples, but not in the reference databases. So it essentially becomes very difficult to find new species and functions that are absent from what is known so far. So I'll start going briefly over some methods for the assembly-based pipelines. So as I was mentioning, assembly essentially means reconstructing longer sequences from the short read alignment. So you start with your clean sequence reads that span 100, 300 base pairs, and you can use tools like Metaspace or Megahit to essentially reconstruct longer contiguous sequences, so usually refer to them as contigs, that will span usually at least 1 to 2 kilobases. This essentially allows reconstructing full-length genes and potentially full-length genomes as well. And the process is essentially like making a DNA jigsaw puzzle where we're trying to find overlaps between different sequences, how they interact with each other, how they align together, and then build this longer skeleton of the genomic data that is in your sample. Now, one challenge with working with and performing genome assembly is that you require a certain level of sequence depth to be able to have a confident and good quality assembly. So in the metagenomics world, one approach that is traditionally used to try to essentially increase your sequencing depth and your coverage is to actually pull samples together before doing the assembly. And that can lead to an increase in the yield of these metagenome assemblies. This process is usually referred to as co-assembly. So essentially you have sample A and sample B, and essentially you join the sequence together, you put them all in the same file before running the co-assembly. Now, it's important to keep in mind that although this process of co-assembly can help, it can allow you to recover low abundant species, it can also create additional challenges. If the samples that you're mixing, so sample A and sample B here, are too distinct from each other and have very different strains, very different species, by joining them together, you might actually be creating more problems and creating more fragmentation in the assembly because you're essentially going to add more variation in the assembly graph that is not going to be able to be kind of resolved. But this is usually a bit of a trial and error and needs to be informed a bit by the experimental design. So if you have samples, for instance, that are from related individuals or even from the same individual but different body sites, you might consider pulling them together. But if you have two samples that are very, very distinct from each other for different studies, different regions, different populations, it might not make much sense to do co-assembly. So then after you get your assembly, the one thing you might be interested in doing is actually be able to recover individual genomes of the bacteria or of the prokaryotes that exist in your sample. This is a process usually referred to as binning, where you're able to generate what are called metagenome assembled genomes, or MAGs for short. So in this example here, let's say we start with a metagenome assembly where there are 3 species present there. So we have the species in green, red, and blue. We can use binners like MetaBat2, MaxBin2, Concoct to be able to essentially tease apart these different sequences into different pots and just have one MAG for each of the species that is present. The way that these binners work is they essentially look at different features in your sequences. The two main ones that are used are the tetranucleotide frequency and the abundance. So tetranucleotide frequency basically represents essentially all the possible combinations of all the nucleotides in your sample. So they look at those, they quantify the frequency of them across a different context, and they use those numbers and those patterns to see which contexts are more related to each other. So essentially, the goal of using tetranucleotide frequencies is to look at contexts that have similar sequence compositions. Now, with the abundance, the idea is to look at sequences that have similar levels of depth. So let's say we have 3 contexts, 2 contexts that have very similar sequence depths and one that has much lower. You would assume that the contexts that have similar sequence depths would be coming from the same species. So that's an additional information that you can use to try to say, okay, this sequence and this sequence belong together, so I'm going to put them in the same group. So if they're green, I'm going to put them in the green group. Now, one thing to keep in mind is that this process of binning is not perfect. So the tools always rely on these big assumptions that these two features work quite well and are pretty reliable, but sometimes that does not work as well in very complex environments. So one way to try to evaluate how well this process is working is to quantify the level of completeness and contamination of a MAG. And this is usually done by looking at what are called single copy marker genes. So in every bacterial species, there are a certain number of genes that you expect to be present and at a single copy. So these are conserved across all bacterial species, and you only find them as a single unit. So you only find them once in every bacterial species. So based on the composition of the single copy marker genes in your MAGs, you can try to estimate how complete it might be or how potentially contaminated it might be. So let's say we have this set of genes that we expect as our ground truth, so the genes we expect to be in all bacterial species. If we have all of them and only these genes in one MAG, then we estimate that will be 100% complete and 0% contaminated. However, if we're missing a big chunk of the genes that we expect to see, then we can, based on the proportion of the genes that we detect, we can estimate that the completeness is going to be lower. And in this case, it will be around 60%. Now, you might run into an issue where not only are some genes missing, but for certain copies of certain genes, you might actually have more than one copy of that gene. And in this case of the MAG, so we're missing the gene here at the end, and we also have double copies of this one. So what this suggests is that not only is this MAG not complete, so it's about 80% complete, but it might also be contaminated. So we might have 2 genomes, 2 strains, or 2 bacteria put together in the same MAG. And based on the fact there's only one gene, then in this case, we estimate the contamination not too high, would be about 10%. So there are automatic tools to try to estimate this. The most common, most popular tool that has been done in the prokaryotic world is CheckM. There's actually a new version of CheckM that was recently released, and it's very reliable for getting these sort of completeness and contamination scores. Then there are other tools that are a bit more catered to non-prokaryotic species, things like eukaryotes, and tools like UCC will work quite well in trying to estimate this level of completeness and contamination. So after you get the level of completeness and contamination, then it's a question of what thresholds do I use? Do I keep the MAG if it has low completeness? Do I keep what value will I use as the deciding factor of whether to use or not the MAG? So there are standards for this that have been published a few years ago. So there's the most common one, the metagenomics space, is the MIMAG criteria, so the minimum information about a metagenome, assembled genome. And they define essentially this criteria for what is a finished, high quality, medium quality, and low quality MAG. Usually, in the metagenomics world, we usually tend to define high quality as something above 90% completeness. In stricter terms, the MIMAG criteria actually requires the MAG to also have the 23S, 16S, and 5S RNA genes present there. But the reality is that in practice, it's very rare, or it's probably less than 10% of your MAGs that will actually have those RNA genes, especially if they're coming from short read sequencing. This is usually because of the fact that these genes are so conserved and they'll be found in so many different species that when you do the genome assembly, you're just not able to separate them out. So in reality, people sometimes adopt modifications of this high quality classification and use a classification called near complete to essentially define what is more than 90 and less than 5% contaminated, but may lack the RNA genes. Usually, looking here at the low quality, usually we don't work with low quality genomes. A minimum threshold of 50% is really what you should be aiming for. And even the medium quality draft, when they tolerate up to 10% contamination, 10% contamination can actually be quite high, especially if one considers that contamination estimates usually are underestimating the actual level of contamination in the genome. So what you really want is about 5% contamination, and then with completeness, you can either use 50 or 90 as kind of your threshold, depending on how strict you want to be. So in any of these MAG studies, you're going to reach a point where you generate all these MAGs for samples, and at the end, you're going to have a pile of hundreds or thousands of MAGs to work with. And then the question that you're going to ask is, should I de-replicate? Should I remove redundancy among our MAGs or not? So redundancy in this case means if you are analyzing independent samples and generating MAGs for all the samples in your study, it's inevitable that at the end, you're going to get MAGs that represent the same species or even sometimes similar subspecies within that species. So then it becomes a question, do I want to kind of collapse that diversity and reduce the redundancy, or do you want to reuse all the genomes and all the MAGs that I've covered? And it's a bit of a debate whether you want to do that or not. And there are advantages and disadvantages to it. So why should you actually replicate? Why should you remove that redundancy? So you should remove that redundancy if you're thinking about essentially doing analysis at the species level. So if you have multiple strains from the same species, it's definitely beneficial to collapse that diversity to make the analysis more manageable. It would also allow you to speed up downstream analysis. So most of the tools that we use scale linearly with the number of genomes that we analyze. So the more genomes you want to process, the more time you're going to need, the more resources you're going to need to actually process these analyses. So by collapsing the diversity of something like a species level, you're essentially making your analysis much easier, more streamlined. And at the same time, if you want to create a reference database that you're going to map against, it can actually be to your benefit to actually do some sort of de-replication to remove a bit of redundancy. Because when you're mapping the reads to a reference databases, if you have highly similar genomes in your reference data, that's going to create problems in the mapping. Because if you can imagine, if I have one sequence that I want to match to a reference database, if that sequence is representable multiple times in a database because there are too many similar species, the mapper is going to have a hard time deciding where do I want to fit this one. So if you reduce that redundancy to some extent, it's going to be much easier for the mapper to find sequences that match uniquely to a genome in your database. And then on the other side, why would you not want to de-replicate? So this is really when you want to do some sort of pan-genome strain level analysis. So if you really want to take into account all the intra-species diversity within your samples, within your MAGs, and then it might not make as much sense to de-replicate. You might still do some very light de-replication, so remove, for instance, identical genomes, things that are 100% identical, but maybe you will not want to collapse that further into a species level. So you are able to create more complete genome catalog and obtain the pan-genome of everything. So there are various tools to do these sort of de-replication. D-rep is the most common one, is the one that I've used the most in our studies, developed by Matt Ohm, which I'll actually be mentioning him quite a bit because he has some other insights into the reference-based methods. And what D-rep does is essentially you give it as input a list or let's say a number of mag files. So these will be represented in a FASTA format. And what D-rep will do is it will look at all your mags and then we'll select the best quality version of that mag as the representative based on whatever threshold you define. So let's say I'm dereplicating at an estimated species level, which would be 95% nucleotide identity. What this dereplication tool will do is it will group the sequences that are within 95% nucleotide identity and then select the one that is more complete and potentially less contaminated. So let's say we have two species in our collection, species in blue, species in orange. We have three mags for the species in blue, one much better than the others. One is actually quite a mix of two different species. At the end, D-rep will only select the best version of that one. The same for the orange. We have one orange here that is a bit fragmented, another is highly complete, and then the middle one is kind of a mix between the two. And then at the end, you will get the best version of those two mags. Here we're talking about strain level, but this essentially could be applied to whatever threshold you define. So this could be strain, species, whatever you threshold you use. Then once you get your kind of dereplicated set, then the next obvious step is actually trying to determine what are the actual bacteria that I'm working with. What are they? What names could I give them? How do they match existing reference database? And to be able to do that convincingly, one of the kind of gold standards in the field is this Genome Taxonomy database, or GTTB for short, which essentially currently includes more than 400,000 genomes from 80 species. And it is actually regularly updated to accommodate the consistent release of new mags and genomes. So this is one of the main important things in the metagenomics world, since new data is constantly being released. Using tools that keep up to date with new developments, I think it's extremely imperative. And one important thing in the taxonomic world is that, so historically, before things like the GTTB came about, taxonomy was a bit of a mess in the prokaryotic world because people were using a combination of genomics, biochemistry, morphology to label and classify different bacteria. But then what GTTB did is actually build a taxonomic framework that only relies on genomic information to do that, which essentially leads to a much more standardized nomenclature. So you can see here, I really encourage you to have a look at the original paper, Nature Biotechnology in 2018, to look at what they did. But essentially you can see here in this graph, so on the left, this is how, for instance, the Coccidia-Alice order was all grouped together. And then here on the right is what GTTB actually did and separated this order into multiple suborders. So essentially before, we were grouping things together that were actually very distinct. And having now this GTB framework allowed us to kind of build a more robust separation and classification of the bacteria and prokaryotes that exist in our samples. Okay, so now moving on to reference-based methods, I'll go briefly over what they mean and kind of some of the advantages and uses that we can get out of them. So there are different, we can kind of classify the reference-based methods into different types. And this, again, was a document taken from a document created by Matt Olm, from the manual of this tool that he developed called InStrain. And there, Matt actually discussed a lot of these concepts quite in depth, so I suggest you have a look. But essentially, briefly, reference-based methods can be kind of classified into three types. So we have the k-mer-based methods, gene-based methods, and genome-based methods. K-mer-based methods essentially mean that they're using sequences of length k, so usually 35 base pairs, to find exact matches between your sequence trees and whatever is in your database. Since these sequences are very short, then these tools tend to use a classification system using the lowest common ancestor to try to predict what is the species or the genera or the taxa that exists in your sample. And this is what tools like Kraken2 do. Then you have more of a gene-based analysis where tools like Metafline and Motus use a set of marker genes, so genes that are specific to specific taxonomic clades, and then you're mapping the reads to these genes, and it uses the information onto which genes it mapped to try to derive what is the taxonomic composition of your sample. And then you have a genome-based analysis, which essentially means that in your database, you don't have just the genes, but you actually have the full genome of your species. So you're mapping the reads to the full length of the genome. You're actually using the paired end information of your reads, and you can calculate more robust statistics about the breadth of coverage, depth of coverage, and depth to try to then estimate what is the prevalence or potentially abundance of your species. There are advantages and disadvantages to all the different methods. K-metre-based analysis, clearly the main advantages of these methods is their low compute cost. They're very fast and they require much less compute than other methods, but they have severe disadvantage in terms of the accuracy and the fact that you're very limited in how much information you're able to extract from them. In gene-based analysis, they generally have a high compute cost just because there are so many genes that are used for different taxonomic clades. And compared to genome-based analysis, they tend to have lower detection accuracy. Since you're only mapping to marker genes, you don't have the full breadth of the genome to do robust statistics and calculate breadth of coverage to be able to reliably say, okay, this species is present, this species is not present. So it generally provides lower resolution than a genome-based analysis. And a genome-based analysis is sort of my preferred way, and this is kind of what Matt also argues, because it provides a good balance in terms of compute costs. It's not highly costly, but it's also not super quick. But it allows you much more flexibility in terms of understanding how well your sample is represented in the reference database, how much breadth of the genome is represented. You can use that to actually be much more confident in your detection accuracy. You can even more reliably estimate what proportion of the sequences do not match reference data. Since you're matching to the whole genome, since you map to the whole genome, whatever does not match are true unknowns, things that are missing from the reference database. Whereas for the gene-based analysis, you might not be matching to one of the genes there because it just represents an accessory gene or a gene that is not part of their marker gene database. So once you have your mapping results, so the end goal of using any of these tools is to have ultimately this sort of matrices. So a sample by species matrix where you get sort of the abundances of the species across different samples. So one of the things that is very common to do in the microbiome world is then to calculate diversity metrics from that data. And you can do things like alpha diversity and beta diversity. So alpha diversity essentially means looking at the diversity within each sample individually. So looking at the level of richness and evenness, so how many species you have in your sample and how evenly distributed they are within your sample. And you can use a metric like Shannon and Simpson to estimate that. But then you have beta diversity, which is essentially comparing the diversity between different samples to see what are the differences between them. So in terms of the overall composition of the microbiome, how different is this sample from this sample? And you can calculate that using distance measures like Bray-Curtis and Jacquard. So then one of the main things that you might be doing if you're doing a human microbiome study is actually try to understand how well does my microbiome composition in my samples correlate with any sort of clinical or phenotypic data that I have available for those samples. So let's say we have a study where you have a control group and a case group. You might be interested in trying to correlate what are the species that are particularly associated with the control group versus the ones that are associated with the case group. So in order to do that, I'm going to essentially just briefly mention two approaches that we are actually taking in the team and represent how we can try to answer these questions. So one is using machine learning. So machine learning has become quite a big popular methodology in not only the metagenomics world, but in all biomedical sciences. And what machine learning is really able to do is to try to see how strong is the association between your microbiome data with a particular clinical phenotype, something like health or disease. So in our study, what we usually do, so we take our sample species abundance matrix, we do first a batch correction. So essentially, if we're doing a meta-analysis where we're comparing multiple studies or even within the same study, but have different batches from different library preparation methods, etc., we can essentially correct for those technical variations. And it's important to do that because we know that those technical variations and the effect of study when you're comparing multiple studies actually most of the time is even more pronounced in the actual biological signal. So you do definitely need to take those into account. Then we take the correct abundance data and we can use supervised machine learning models, things like logistic regression, random forest, and gradient boosting to try to estimate how strongly does the microbiome relate with that phenotype. And this can be evaluated with a metrics like the area under the curve or area under the precision recall curve. I actually have linked here a pipeline that I put together to do this. Is actually based from a pipeline from Patch Plus and Microbiome, which I just adapted for our own study purposes. So you can have a look there if you want to try and play around with some of these methods. So the machine learning is just to have kind of a broad answer of, is your microbiome strongly linked with that phenotype? and then you'll get a metric. But then you might be actually interested in understanding what are the species, what are the features within your microbiome that most strongly relate with health and disease. And this is when you do this differential abundance analysis, and you can use a combination of different tools, things like Maslin on Aldex that use a combination of either linear mixed effects model or generalized linear model to try to answer these questions. The advantage of using these types of models is that you can provide additional covariates, so additional information you have to control for potential batch effects or potential confounders. So we know that things like age, continent, and again, batch will have a strong influence on the microbiome. So these methods allow you to provide us as covariates, so then you're able to tease apart what is actually associated with the health state. So here I'm just showing an example. So this is not a paper from Microbes, it's just a paper released a few years ago where the authors did a meta-analysis of different studies, and they're essentially trying to find common markers that were coming out of these studies. So each study here represents one group of samples linking to a specific disease. So we have things like colorectal cancer, IBD, type 1 diabetes, etc. And you can see here the area under the curve metric that they retrieved. So some diseases are strongly related with the microbiome, where others not as much. And then here you have the number of genera or taxa that came out as significant. And then on the right, you can see kind of which direction that taxa is. So you could have taxa that are enriched in disease, so this is what they call pathogen enriched, or enriched in health, which would be the beneficial depleted. And you can then also compare across different studies and look at what is the profile of all of these. Okay, and just one final thing. So I'm not going to go into details here, but one thing you might be interested in doing is once you have your candidate set of species and you have identified reliable biomarkers, is to do a functional annotation of your species. So let's say you have species that are associated with health, species associated with disease. You will take your genomes, do some sort of gene prediction on the genomes, and then use a wide range of annotation database to try to estimate the functional capability of the genomes. And this is where you might start to be able to build hypotheses on why you're seeing some of these genomes associated with certain phenotypes. And you can use things like eggnog, KEGG, anti-smash, gut smash, to essentially have robust understanding of the primary, secondary metabolism, all the pathways that exist in your sample. Again, I'm plugging here another pipeline I put together, so called Genome, that actually does a lot of these annotations independently. So do have a look there if you wanna play around with some of this. So in terms of main conclusions, I hope I was able to show that there are many metagenomic methods available and approaches to analyze microbiome data. Reference space usually are the easiest and will give you a roughly quick answer of what is there, but you do want to consider assembly-based methods if you're working with very unusual samples or things that might not be well represented in reference databases. But ultimately, it's just important to really choose the right approach and statistical method for the data and question in mind. So do spend some time thinking about that. As a last thing, I have I have left here just a number of papers and tutorials you can have a look. So we released a paper a few years ago. This is essentially a protocol paper on discussing a lot of the concepts that I talked here today, just showing step-by-step how you might be able to recover proteotic genomes from host-associated data. There are other review papers that I encourage you to have a look that discuss a lot of these methods and other approaches. And there are even tutorials that will give you step-by-step examples. So there's the Meta pipeline, which does the binning, so it generates mags. And we also, I used to be part a couple of years ago of the Magnify team here at EBI, and we actually put together some training materials that are freely available. You can have a look. That will also go through some of the concepts that I discussed here. So as mentioned in the beginning, I'm based here at the Department of Veterinary Medicine, and so my lab is based here in the department. I lead the Microbiome Function and Diversity Group. Now let's hear the contact information just in case you have any specific questions I will not have time to address here today. Happy to answer through email or any means that you're able to contact me. Thank you all for being here, and I'm sorry for the cough. The tea helped a bit, but probably not too much, and I'm happy to answer any questions that remain.
Thank you very much, Alex. That was a very informative talk, and I'm sure, and in fact, I can see that many people in the chat are keen to learn about this and also to ask lots of questions. I think I'm going to have a look through the questions based on the sort of the thumbs up to have a look what people are interested in seeing answered. one of the ones that I just wanted to mention was that someone asked about if you could recommend a book for studying metagenomics. And obviously you had some resources on your previous slide. And just to mention that we will be sharing the recording and the slides, and you'll get an email with those tomorrow so that you can go back to look at those links that are shared there. And yeah, someone's saying how to access a link. So yeah, we'll make sure you receive all of those tomorrow. And I'm sure you can sort of learn from any of those. I didn't know if you had any other suggestions on that, but I'm sure those are good.
So, I mean, books are not usually the most efficient way to get things across. It's just because of how fast the field moves. You really want to look at published works, so things like review articles or manuscripts or research articles, because books tend to take a long time to get published, and then by the time they're out, they probably will be already outdated. Books are good for more conceptual ideas like in microbiology, but for actual computational metagenomic methods, sometimes it's hard to find like a good book. So yeah, my suggestion would just be go through some of these resources I put here, which I think will give you a good basis.
Perfect, thank you. I've also just shared in the chat a link to the course materials from a metagenomics course that runs from the EBI every year. And we make those materials available so that everyone can work through them in their own time as well. Okay, so having a look at the questions in the Q&A box, we have the top one. How can one tell if some of the genetic materials extracted from the sample are not introduced through water or food ingestion and not actually part of the microflora?
Yeah, that's an interesting question. I guess things that would be just transient, things that you are ingesting in the moment, will tend to be rarer or less prevalent in your population. So in most cases, so in the gut microbiome where we don't have as much issues with contamination from like laboratory kits or things like that. But in general, you want to be mindful of things that you detect at a very low prevalence in your population, at a very low abundance. So if you have 100 samples, you only detect certain sequences, certain species in one or two samples, you might not pay a lot of attention to those because it's a higher risk of them being either contaminants or things that are very transient in your population. One thing I'd like to mention, there was actually a recent paper coming from Sean Gibbons' lab, where he actually developed a tool to quantify or to try to estimate the food, like the composition of the food that people are ingesting by looking at the DNA composition of foods that are, that have genomes for. So you can use tools like that. So you can have a look at what the work he did to help with that. So you might be able to use that tool to sort of distinguish a bit more, okay, what are the sequences, where the DNA coming from, like food specific sources versus other that might be more microbial or things that you might be interested in.
Thank you. Having a look through some questions. So the next one we have, what's the difference between the human genome analysis and microbiome analysis? They share some similarities in workflow, but some differences as well.
Human genome versus human microbiome analysis.
Yes.
So these will be very different because human genome, you'll be looking just at the DNA of the host. So in case it would be like an eukaryotic genome where you're trying to look, at variations in the host, maybe doing some GWAS studies looking at different populations and how they relate with different risk factors. But the microbiome, you're looking at the bacterial and the prokaryotic component of the host. So it's going to be very different approaches. They're both the DNA, but they work very differently based on the assumptions and based on the type of material that you be working with.
Thank you. Sorry, my mouse just threw me right down the list of questions and threw me off there. So we have here, why should you perform metatranscriptomics if you can also obtain functional information with metagenomics? I'm getting my metas mixed up here.
So the value of metatranscriptomics is that you are able to investigate what is being actively expressed by your genome. So the genome is sort of the blueprint for what the bacteria can do, but it doesn't necessarily tell you which parts or which genes are being more actively induced and stimulated. So the metatranscriptomics will tell you, if you, for instance, you have a genome for a gene, you have the sequence for a gene, but then when you do metatranscriptomics, you don't get any transcripts for that gene. that probably means that gene is not being utilized or being active in that particular sample, in that environment where you isolated it. On the other hand, if you detect huge number of transcripts for a certain gene, then you might speculate that that gene might be particularly important for that environment because it's highly expressed, highly active in that environment. So it does provide a bit more of information on the activity of the bacteria rather than on the potential of what the bacteria can do.
Excellent. Look down the list. Someone asks, is it possible that by throwing out reads that map to reference host genome, we throw out microbes that are part of the reference as retroviruses or intracellular pathogens that weren't removed from the reference?
Yeah, I mean, in the case of viruses, it could be possible. With bacteria, I guess this would be less likely, unless the actual genome is contaminated. So whatever reference genome you're using for some reason became contaminated with prokaryotic sequences. In general, from my experience, the proportion of reads that are mapped to your kind of human genome that are kind of false positively mapped, that you would want to keep that are mapped to human genome, would be a very, very low portion. You rather be more conservative and actually try to map as much as possible, and then risk is still having human sequences in your sample, and then those human sequences mapping to the bacteria and then giving you erroneous conclusions. So I tend to err on the side of caution of this. And I admit that maybe for viruses, it might be more problematic with things like retroviruses. That could definitely be a consideration to make. But for prokaryotic genomes, that's not usually a problem.
Okay, thank you. You talked a little bit about machine learning towards the end there. And someone asked for your use of machine learning, was there a minimum number of samples that you needed to use?
So, I mean, the question of the number of samples really depends on the effect size of your, the relationship between your microbiome and your phenotype. So if you're looking at diseases like Crohn's disease, the effect size is huge. So I would say even with 50 samples, you are able to detect a very easy signal. But if you are talking about things that have more of a direct role, there are diseases like, for instance, multiple sclerosis or Parkinson's disease that do have an association with the gut microbiome, but is not as strong. You definitely would need to increase the sample size. Usually what you want to do is do some sort of statistical calculation to estimate what is the minimum number of samples you need to achieve the statistical power to detect the difference if there is one. And there are tools and methods out there that allow you to do that. I haven't put them up here, but if you just do a Google search on statistical power analysis in microbiome data, there are a couple of papers that have discussed that, and we'll give you some information how to do that.
Sounds good, thank you. Someone asked if you could elaborate on the differential abundance methods that you mentioned. Which do you think is perhaps more appropriate for the shotgun metagenomics data sets?
Yeah, so unfortunately, this is a very debatable answer because there are tons of methods to do differential abundance. And there has been a paper coming out a couple of years ago that tested around 30. And they show that in general, although the strongest signals do agree across methods, they all carry some inherent differences. And the bottom line is that there's not one method that you can say this is the best and this is the only one you want to use. The general conclusion is that you want to use a combination of methods. That's why we use at least two to see what is common across the methods, and then be more confident in what you define as differentially abundant is actually more robust. Because they are, so for instance, I mentioned Maslin 2 and Aldex 2, and not only do they use different statistical approaches, but they also rely on different transformations of the data. So with Maslin, we're using relative abundance. With Aldex, we're using center log ratio transformations. And for the center log ratio transformations, people argue that is more robust to variations in sequence depth. But some papers then say that's actually not the case. And we do some simulations and show this is not. So there are various conflicting answers. So our position in our lab is that so we try different methods and we find the intersection between them, and whatever comes out as common between the methods will be the ones that we discuss and are more confident in. And I think that should be generally the recommendation for all these types of studies.
Thank you. So just having to say there are a huge number of questions and that there's not going to be a chance to get through them, but it's really nice to see that so many people are keen to engage in the session and ask their questions. So we have here a question asking about microbiomes from people in different regions. And they're asking how it differs between different regions, and if there's a need to take probiotics when relocating to a different region, given changes in diet.
The probiotic question.
Yeah.
I'm always waiting for that one. Yeah, so there is a huge variation. So that's why when I mentioned differential abundance, geographical location, that needs to be kind of a covariate you need to use in your mouse, because it does vary quite a bit. Usually, so when you do these sort of studies, the most important factor that distinguishes your microbiome is kind of the study effect. So if you do one study versus another, and then the second most will be country or location. So people from westernized populations are going to have a very different microbiome from non-westernized. So Africa, South America, Europe, North America, they all have very different microbiomes. So geography is going to play huge roles. In terms of the probiotics question, essentially, the research so far does not support a positive or beneficial role of probiotics in the human gut. I don't necessarily say that it's detrimental. So there actually have been studies showing that it actually could be detrimental in certain aspects. But these studies have also been done in relatively small populations. The bottom line is, I think the research on probiotics is still largely inconclusive and shows some conflicting results. So I would say, if I had to make a recommendation, I would say you don't need to take probiotics. I don't necessarily say that they were going to be harmful, but I don't trust the benefits that people usually proclaim about the use of probiotics for gut health.
Okay, thank you. Hopefully that helps them. And then we had a question here about where samples are coming from. So you mentioned about getting samples from stools for analysis. What other sources are used for getting samples for human microbiome analysis?
So the other approaches, which end up being more invasive, are things like colonoscopies. So people, for instance, if they're doing a screening for colorectal cancer, some studies have actually used that opportunity to essentially sample the microbiome from these individuals and actually do essentially biopsy of the sample and study that. So essentially, this gives you more access to the mucosal microbiome, not what's in the lumen. There are new fancy techniques coming out, things like what is it called? There was a cap scan, I think, which essentially people ingest a capsule, and then that capsule will essentially sample your microbiome at the end. And then when you essentially excrete it, then you're able to get the microbiome that is there and sequence it. But that's still not very popular because there's still some kind of validation going on about the stability and the reliability of these results. Then there are things like intestinal lavages as well. But again, these are all very invasive methods. So that's why, taking stool samples is usually the preferred or the most common method, because it's the easiest for people to get and do these sort of large scale studies. If you're doing anything more invasive, you're obviously going to only be able to do it in a very small number of individuals when there is a medical reason to use this sort of interventions. You wouldn't do it for like a normal health individual, which obviously these methods can carry their own unique risks as well.
I think maybe just one more question here, something slightly different. Someone asks, for metagenomics analysis in general, would it benefit from a single cell perspective and looking at that kind of level?
Yeah, so there have been some work doing single cell metagenomics, specifically in this idea of generating metagenome assembled genomes. So when you're working with a metagenome where you have a huge diversity of species in there, and you're trying to tease apart the mags within that sample, it definitely helps if you have single cell data, because then you're essentially separating beforehand experimentally each bacteria, and then you essentially reconstruct the genome of those single cells. What has been noticed is that the current single cell procedures tend to reduce a bit the amount and the yield you get from the bacteria. So although you get purer genomes, in the end, you actually get more fragmented or less complete genomes. But I do see potential for growth in that area, and that might be an opportunity to sort of circumvent a lot of the limitations that these metagenomic methods have in terms of mag generation. So yeah, look out for further developments on that.
Great. That sounds good. So thank you very much for answering so many questions. Obviously, there are many more, but we unfortunately don't have time for all of them. But we really appreciate all the questions that everybody's asked and for getting so involved in the session today. So that just leads me to say thank you very much to Alex for joining us and for his wonderful presentation. and to everyone for joining in with today's webinar. As mentioned, the recording and the slides will be made available tomorrow, so I will make sure you all receive an email with the details of that. So you're very welcome to come back to them and have a look through, and of course take a look at the links that were in the end of the slides there. I can see lots of thank yous to Alex in the chat, so really appreciate you coming along and joining us today.
Yeah, and as I mentioned in the talk, feel free to reach out if you have any more specific questions. I'm happy to take some time to answer that through email or any other means. Yeah.
Wonderful, thank you. So we look forward to seeing you all at a webinar in the future, and hope to see you at one of the series of webinars as well. Thank you, everyone.
Thank you. Bye-bye.